Kia ora, I'm Murph, properly Dr. Charles Murfield, head of the BHU Future Farming Centre, and this is a webinar of the just released report on soil health and its management. So this webinar covers the entire contents of the report. So Future Farming Centre, let me just move this out the way a fraction. So this is a research centre in New Zealand, based at Lincoln University, although separate from the university, focusing on the sustainable agriculture, such as organic, regenerative, and agroecology, and producing information for farmers and growers. Quick disclaimer, we've made every effort to make sure all this information is correct, but we take no responsibility. This is YouTube after all. If you want to get access to the full report, there's a QR code there if you want to snap that. Website, and also I'll put the link in the description below. And this report and this webinar was sponsored by the Irish Organic Association. Both the report and this webinar is based on a report done for the Irish Organic Association focused in on organic agriculture. This is for a wider audience across all of agriculture. Quick outline of where we're going. We're going to touch on the importance of soil and its health, then look at the soil science revolution that's occurred over the last 15 odd years, and then look at the three soil healths of physics, chemistry, and biology. And we're going to dive into that in quite some detail. And we're going to round off by looking at some DIY soil health tests, stuff you can do yourself when standing in a field. So, the importance of soil. It is impossible to overstate the importance of soil. It's right at the heart of our planetary systems and our biggest planetary challenges, stuff like climate change. The carbon cycle goes through soil. Indeed, soil is the second largest store of biological carbon after the oceans. Biodiversity loss. Soil is the most biodiverse ecosystem on the planet, so it's right in there. Nutrient pollution principally from nitrogen and phosphorus. That's coming from soil. The water cycle, the hydrological cycle, passes through soil. Land system change, how soil is affected by changing from natural ecosystems into farmland and farmland into cities where the soil is sealed, and novel entities. Soil's responsible for detoxifying most of the pesticides we put into the environment. As I just touched on, soil is the most biodiverse ecosystem on Earth. About 60% of all species live on soil, based on this recent scientific paper here. And particularly for the small organisms, the microbes, the majority of them make soil their home, as this infographic here highlights. Soil is also critical to the provision of many ecosystem services. Ecosystem services are the services provided to humanity, our civilization, by ecosystems, by natural systems, and soil has a key role in most of them. It's the foundation of farming. Most of our food is grown in or on soil, i.e. livestock. There's a small amount of food comes from water, fish, from the oceans. Virtually all our food is grown in or on soil. And so therefore farming is the foundation of civilization because after having air to breathe and water to drink, food is the third essential things we require to actually be alive. So farming, feeding ourselves, growing our own food, is the only essential industry. Forget not having smartphones. Forget not having muscle power in terms of livestock for draft animals. It's the only thing we have to do to keep ourselves and our civilization alive. And it's the foundation of farming in that healthy soil grows more crops and pasture. And I'll give you some examples of that as we go through. So basically, healthy soil means you can produce more and make more profit. Also, when you buy a farm, the only thing that's irreplaceable is the soil. All the infrastructure, the buildings, the fences, perennial crops, whatever, shelter belts, can all be replaced. So soil is the primary capital asset of a farm. So when you lose soil to erosion, wind and water, that's essentially capital value blowing away. So you cannot overstate the importance of soil, and we have known this fact since the dawn of agriculture. The Sanskrit scriptures here 11,500 years ago. understanding that our survival depends on soil, and if we look after it, it will look after us, abuse it, and we will cease to exist. Despite knowing this for over 10,000 years, we still far too often fail to implement it. So fundamentally, this is what this report and this webinar is about. Science is also increasingly viewing soil as a thing that is alive itself, not just a structure, a scaffold in which other living things live. And part of that is it's increasingly being viewed not just as a static entity, but a series of processes, a complex adaptive system. The key thing between something that's living and dead isn't the number of nutrients in it. They're exactly the same before and after it dies, but its processes have stopped. And increasingly, energy flow through the soil ecosystem is seen as a key window on those processes. So, coming to the soil science revolution, over about the last 15 years, soil science has undergone this paradigm shift, and it's principally around the primary inputs of organic chemistry and energy into the soil ecosystem and the formation of different types of organic matter. And this is summarized here in this diagram, and this is taken from this scientific paper on the left by Truffaut et al., pulling together this revolution that's occurred over the last 15 years. On the left-hand side here inside this blinking circle is how we used to think soil organic matter was formed. Stuff dies, leaves fall on ground, plants die, animals die, there's manure and other organic residues. This is fragmented by some of the larger macrofauna in the soil, stuff like worms, beetles, springtails. Then the microbes, the bacteria, the fungi, the archaea hook into that fragmented organic matter and reduce it further, and it becomes particulate organic matter, POM. And instead of this being the main source and route by which organic matter in soil is created, SOM, soil organic matter, SOM, only about 20% of SOM comes down through this route. And it's decomposed pretty quickly, most of it within a few months, but a few years to a few decades at the very most. And humus doesn't exist. Humus has been this material that's described as recalcitrant. It resists further decay. And it was often thought that the tough materials, the lignified woody materials, that as they decomposed, they were converted into humus. And for many, and particularly in things such as the organic agricultural movement, humus was viewed as the foundation on which soil health was based. And it was considered to last millennia, centuries to millennia. But when soil scientists using new tools such as CT scans and synchrotrons, intense light beams, directly look at soil, they cannot find humus. It doesn't exist. Indeed, it doesn't even make sense. You start with these large pieces of dead organic matter that are fragmented, then they're attacked by microbes. And as these pieces of organic chemistry get smaller and smaller, somewhere this big recalcitrant humus molecule appears. And it's kind of like, where did that come from? And what is it about the arrangement of the carbon, oxygen, hydrogen, and other nutrients in humus that makes it recalcitrant, resistant to decay, whereas everything else made up of exactly the same chemical elements is decayed and mineralized quite happily? We now know that humus is an artifact of the chemical alkali extraction methods used last century. The new stuff's over here on the right-hand side. Plants put out through their roots between 10 to 40% of the photosynthetates they make from sunlight as root exudates. These are simple soluble compounds, sugars, proteins, oils, and these feed a vast density and diversity of microbes that live around the plant roots in what is called the rhizosheath and the rhizosphere. The microbes return the favor by providing the plant with nutrients, water, protection from pests and pathogens. Different plants produce different exudates that favor different microbes. Indeed, plants can even change the exudates they make when they need assistance from particular microbes. For example, if the soil is going dry, they will feed fungi, particularly mycorrhizal fungi, that can extract water more efficiently than they can. So this is starting to tell us what's driving this system is a diversity of plants maximizing photosynthesis. Again, that's a proportion, that's a percentage of photosynthesis. So the bigger the plant, the more leaves it has, the more photosynthesis it can undertake, the more the total amount of root exudates it will produce. So the soil microbes that are living in the rhizosphere, feeding on these root exudates, they are consumed by other organisms, amoeba. They are attacked by viruses, and viruses have desperately not realized the importance of viruses in the food ecosystem. When a microbe is attacked by a virus, a bacteria, an archaea, the viruses multiply inside them, and then the bacteria explodes. And this produces a fine shrapnel of organic remnants of that bacteria. And bacteria, apart from viruses, are the smallest living thing. Viruses kind of half being alive, half not being alive. And that produces quite a different result in terms of the distribution of organic matter in the soil compared to a bacteria being eaten by, say, an amoeba. And it is those tiny, tiny fragments of organic matter that then get stuck onto the surface or inside soil minerals, the sand, but particularly the silt and especially the clays. They can go inside the clays. And by being attached to these soil mineral particles, they're protected. And that protection means this organic matter can potentially last for decades, centuries, even millennia. And this is called mineral associated organic matter with this hideous acronym MAOM or MAOM, which we're not even sure how we should be pronouncing. But this is now the most important form of organic matter in the soil. Some POM is also transferred into MAOM through microbial decomposition as well. So this has some key implications for how we farm and look after our soil. Fundamentally, it's a diversity of living plants with lots of photosynthesis going on, which is going to maximize root exudates, that's going to power up soil biology, the rhizosphere, the microbiology living around those roots. That maximizes soil health. Our old ideas that it's organic inputs such as plant residues from cover crops, manure and compost, they play really quite a minor role in this. It is living roots that are key here. So what this means is our standard high school science view of plants is basically upside down. That kind of standard view is plants take up water and nutrients through their roots, and then they push that up and use that to build their foliage. What we have is plants taking energy from sunlight and combining that principally with carbon dioxide from the atmosphere. Plants are about 45% carbon, 45% oxygen, and 6% hydrogen. And a whole load of that oxygen is also directly coming out of the atmosphere. And some of the oxygen and the hydrogen are coming out of the atmosphere as rain, which is being absorbed by the roots, but it is coming out of the atmosphere. So that means some 96% of a plant's biomass originates from the atmosphere, not from the soil. So what's happening is that carbon, oxygen, and the hydrogen from the water are being combined into organic chemistry, carbohydrates, and then proteins and fats and oils, and they're being pushed down the plant into the roots and then out into the soil. So plants are actually pushing out into the soil far more chemistry than they are actually taking up from the soil. So I came up with a little aphorism to describe this new soil science revolution. Humus is dead. Long live Ma'om. Think about it. It works on multiple levels. So we're talking about soil health here, but we haven't actually defined it so far. And this has actually been something of a contentious issue in soil science over the decades. But there is now pretty much a general agreement that there is such a thing as a healthy soil. And we have this commonly used definition by Doran and Zeiss here, the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And I kind of go, yes, that's kind of helpful, but I also think we can even get simpler here. Just think about your own health. Are you fit and active? Do you exercise? Do you have a healthy diet with lots of plants in it? Or are you unfit? Do you sit on the couch watching sports and eating burgers? It's that kind of simplicity. A healthy soil is basically how a soil that is functioning as it would under a natural ecosystem like a woodland or forest. And soil scientists commonly break down soil health into three subdivisions: physical, chemical, and biological. And ecological, biology and ecology are different things, but here we just lump them all together. So physical is structure, aggregation, chemical is basically just nutrients and pH, and biology is all the living things that make soil. And I use this metaphor of a three-legged stool, where each of the legs is one of the soil healths, and the seat of the stool is the energy flowing through the soil ecosystem and powering it. And the metaphor of a three-legged stool is if one of your legs is short, it's not a very good stool, and you can fix it by shortening all the other legs, the other two legs, off to the same level. But now you've got a short stool, so it's not as good a stool. And the metaphor is if you've got one soil health that's not very good and you try and fix it by reducing the health of the others, your soil health overall declines. But more importantly, if one leg is short, you can't fix it by lengthening one or the other, or both of the other legs. You now have a very wonky stool. And the metaphor here is, say, biological health isn't as good as it should be. You can't compensate that by increasing, say, chemical health. That simply doesn't work. You have to have all three healths working together because they all support each other. Physical health supports chemical health, which supports biological health, which creates physical health. They are all interlinked. So all have to be got right together. And just an example here, and we will come back to this in the DIY soil health tests at the end. Healthy soil on the left, this is taken from here at the BHU. From a piece of uncropped ground, bit of thatch of grass on the top there. You can see it's nice and dark. The darkness comes from carbon as organic matter. So organic matter is around 40 to 45% carbon. Carbon is black. It's got a nice crumbly structure, lots of different sizes of those crumbs, the aggregates. There's lots of roots in there, and we can see a couple of earthworms on the right-hand side. This unhealthy soil taken from the middle of a trackway, just a few meters from the one on the left. You can see it's a lot paler. It's got less organic matter, less carbon in it. It's got a blocky structure. There's big pieces in there, few little crumbs, few signs of roots or earthworms in there. So soil physical health is all about structure, that it's well aggregated. And these aggregates are these little crumbs. When you gently tease a piece of soil apart, the aggregates are the bits that don't fall apart any further. And another analogy is soil should be like a bath sponge, basically a whole load of holes, tubes to be more precise, so that are continuous. And about 50% of a healthy soil should actually be pores, empty space, if you like. And this structure, this porosity, is what allows water and gas to exchange between the soil and the air. So oxygen and carbon dioxide and a range of other gases, and water vapor and liquid water as well. And the stability of these aggregates is being shown to be, through a huge amount of science, a really good proxy for overall soil health. So if you have good aggregate stability, all your other soil healths are likely to be good as well. And if you've got poor aggregate stability, all your soil health is likely to be poor. In terms of managing physical health, there's three things that destroy soil structure. First up, that lack of living plant roots producing exudates. And then it's compaction, and then it's tillage. And here's a classic example where people think the health of that soil is going to be really good because it's covered in mulch. But that mulch, some form of bark chips here by the look of it, A, has few nutrients in the likes of bark chips, but B, it's entering the POM cycle, so it decomposes very rapidly, and it feeds none of the microbes that live off plant root exudates. There are some plants here, there's a tree and a few little small shrubs, but there's lots and lots of empty space here. So that soil is literally dying from a lack of plants and a lack of plant diversity. These are all woody species, growing in a temperate region, so they're likely to be dormant over winter, and a dormant plant produces no exudates. It's literally asleep. So for half the year, there are no exudates being produced. The soil is probably more healthy under that mown lawn around this mulch bed than the mulch bed itself. Compaction, we've understood compaction for centuries, if not millennia, going back to the very dawn of agriculture. And if we are going to farm land, some compaction is inevitable. Even if we're just managing stuff with hand tools, us walking on soil compacts it. And I'll show you some data later on showing you can get 4 times the yield under a healthy and uncompacted soil versus a compacted soil. So minimizing compaction isn't just about environmental stuff, it makes very good business profit sense as well. And for anybody involved in farming will be very aware that compaction is worse when soil is wet to moist. Dry soils, particularly clays and silts, can sustain quite a lot of weight on them before they get compacted if they're dry. But if they're wet, then they get very badly compacted. So staying off soil when it's moist to wet is becoming increasingly important, both environmentally and from a business economics perspective. And livestock, even really small stuff like lambs, cause significant compaction at the soil surface. What's happening is I like to say they are squishing the macropores. And for non-soil scientists, macropores is a bit of a confusing concept because it's soil pores that are larger than 0.075 millimeters, about 0.003 of an inch, which is about the diameter of a human hair, which most lay people think is fairly small. There are a whole load of soil pores, the micropores, that are smaller than a human hair, and they actually represent quite a significant proportion of the total soil pore space. What's happening is when soil is moist and the livestock's walking on it, they just very gently compress that top few to 20 centimeters and squeeze these macropores out of existence. And the little lambs are only squishing the top few centimeters, an inch or so of soil, whereas a big bull is going to compact down to 20 centimeters, 8 inches, or possibly even more, depending on the soil texture and the moisture levels. And what this means is the macropores are the main pores through which soil breathes, i.e., exchanges gases such as carbon dioxide and oxygen. It's also the main route by which water infiltrates from rain and irrigation into the soil. So essentially, you lose the ability of soil to breathe. For water to infiltrate, the soil is going to go anaerobic. And particularly if the soil dries when it's in this compacted state, that compaction gets locked in because plants and soil microbes cannot rectify that compaction when soil is dry. Machinery compaction is worse because it goes deep. And two rough rule of thumbs here. Tire pressure determines the density of compaction. So if you took a piece of soil out, what the density of that soil would be. And the heavier the machines are, the deeper the compaction goes. And we've got some examples here. The sources for this are in the main report. A tractor that's got 300 kilos per wheel. So this is basically what an old Massey Ferguson 135 would be exerting on the ground. 300 kilos per wheel, not 300 kilos per machine. About 900 pounds. That goes down to about 30 centimeters, i.e. the plough layer, the A horizon of the soil. So that's reasonably deep. That's 12 inches a foot. 1,000 kilo tractor, so the total machine's around 4 tons. That's going down to 3/4 of a meter, so getting on for nearly a yard deep. And a 4-ton tractor isn't necessarily a terribly big tractor in many situations now. 3,000 kilos per wheel, so this is a big tractor by modern standards. That's going down to a meter, so over a yard deep compaction. And this is getting past the point where we can use things such as rippers and subsoilers to actually reach that deep using tractors and relieve compaction at that kind of depth. You're really getting into big caterpillar crawler type machines to reach down that deep into soil. And then the one on the right at 8,000 kilos is a half-loaded combine harvester. And that is going down over a meter and a half. So this is nearly two yards. We really lack machinery in agriculture that could go to that depth to relieve this compaction that's being created at depth. And there's a wide misunderstanding that it's the 10th time you drive across a piece of ground where the compaction's really mounting up. 75%, three-quarters of compaction occurs in the first pass, not the second, not the third. It's all the damage is done. Most of that damage is done on the first pass across the field. And so this is the basis of approaches such as controlled traffic farming and vegetable beds in vegetable production to use the minimum amount of ground and stick to the same wheelings at all times. What this means is those wheelings get particularly badly compacted, so they're very poor at growing crops, but the rest of the field is uncompacted and therefore grows crops far better. What this also means is compacted wheelings mean you can often get on a paddock when it would be too wet for operations otherwise. And this is taken to its logical extreme in gantry tractors. The Nexstat gantry tractor here has gone out to exceptionally wide wheelings with an enormous machine on enormous tires that's causing a huge amount of compaction in those wheelings, but that is where all the compaction in these systems lies. So the vast majority of the field is now completely uncompacted by machinery. Key approaches to minimizing compaction. This isn't new stuff. We've been banging on about this for decades, if not a lot longer. Minimize the passes of machinery on field, so single pass cultivators or moving to min-till and no-till. Use the lightest machine as possible. Of course, there are always practical challenges here. There's only so many tractors on a farm. There's so many different sizes, so invariably bigger tractors get used when a smaller one would be able to do that job. Keep your tire pressures down, and the tire manufacturers are increasingly coming up with new tires that work at even lower pressures than we already have, and stick to those same wheel tracks. Whatever farming system you're using, stick to the same wheel tracks. Tillage, or if you speak British English, cultivation. All tillage is harmful for soil. It damages soil and soil biology with it. A rough rule of thumb is the more energy used to undertake that tillage, the more damage being done, because simply more energy is being put into destroying soil structure. So draft implements such as springtime cultivators are going to do less damage than PTO implements, particularly things like rotavators. The exception here is ploughing. Ploughing uses a lot of energy. It burns a lot of diesel because you are essentially flipping the entire plough horizon of the field upside down and moving it sideways by a plough width. And that inversion is exceptionally bad for soil health. So we need to minimize cultivation, tillage, but at the same time where extensive soil damage has occurred, for example, from livestock in fields that are wet over winter, it's turned to slurry and it's now set hard. Some judicial use of tillage to break up that initial damage and allow plants to re-establish in there, and for the soil biology, the microbes to get in there and start fixing that damage. Fundamentally, tillage cannot fix soil damage, physical soil damage. Only biology, plant roots, and microbes can fix the damage. And as an example of physical damage, this research here from New Zealand, from Southland down the bottom end of New Zealand, this is a temperate farming system equivalent in Europe to, say, the south of the UK and across to Denmark, climatically. In arable and vegetable systems, they measured the aggregate stability, which, as I've already said, is a really good proxy measure for overall soil health, including physical soil health. So the scores with the lower aggregate stability are going to be more compacted, and vice versa. The ones with a high aggregate stability score are going to be less compacted. And there is about a fourfold increase in yield as you go from the most compacted, lowest aggregate stability scores here up to the very highest ones. So that very clearly demonstrates the economic value of soil health, if nothing else does. And then moving on to chemical soil health. Conceptually, very simply, is basically having your nutrients and pH at optimum, the infamous Goldilocks zone, not too little and not too much. Indeed, the not too much bit is becoming increasingly important. Too many nutrients will actually reduce yield, and too much nitrogen and phosphorus is a massive environmental pollution risk. So we really need to be increasingly making sure we are not oversupplying nutrients. And I will return to that topic as well. So I don't want to kind of just do the standard soil test fertilizer thing. I want to look at chemical soil health from three different perspectives. That is stoichiometry, which is the science that studies the ratio of the chemical elements of life, but also ratio of chemical elements and all kinds of other things as well. Nutrient cycles, both at the soil and global level, and the law of the minimum, Liebig's barrel. So stoichiometry. All living things, plants, animals, bacteria, have roughly the same proportions of the chemical elements, the nutrients. We are all mostly carbon, oxygen, and hydrogen. Plants are about 96% carbon, oxygen, hydrogen. Animals are about 90% carbon, oxygen, and hydrogen. But we don't typically think of these as fertilizers because they are replenished by the atmosphere, which I will come back to shortly. So what we typically think of as fertilizers, nitrogen, potassium, calcium, etc., NPK, only represents about 4% of the total dry biomass of a plant. We also here have the essential elements and the beneficial elements, and I will come back to those in a second. Because all things have the requirement to have roughly the similar proportions of the chemical elements, if we've got our soil nutrients correct for plants, that means we're going to have the right amount of nutrients for all soil biology, the bacteria, the fungi, everything. We talk about macro and micronutrients. Macronutrients are needed in larger amounts, typically around 1% or more. And I've put carbon, oxygen, and hydrogen there to reinforce the fact that these things are nutrients. They are chemical elements life needs to survive. And then the micronutrients are typically needed in less than 1% amounts, down to 0.0001 kind of percent amounts. And those essential and beneficial nutrients. Plant science, botany has developed compared to, say, animal science, zoology, has developed a really strict definition of what essential nutrients means. It means if it's absence, the plants will die. Not that they'll just be sick or something, they will die. And this has flowed through to legislation such that only in many jurisdictions, only essential nutrients can be sold as fertilizer. But we know there's a whole bunch of non-essential nutrients. Plants won't die if they don't have them, but the plants will actually grow an awful lot better if they do have them in the right amount. Things like silicon. So, increasingly, I think we should also be testing not only for the essential nutrients, but for these non-essential nutrients or these beneficial nutrients. And I know I've asked here in New Zealand, some of the soil testing laboratories will test for a number of these beneficial nutrients as well as the essential ones, if you ask. Building on that, animals, although we share the same rough proportions of nutrients as plants, we actually need a larger range. Plants need some 20 essential elements. Animals need about 30, just shy of 30. Classic example here in New Zealand is selenium. Many of our soils here are deficient in selenium. Often only pasture land, which has grazing animals on it, is tested for these animal essential elements on the basis that cropland is just growing crops. So the plants don't need these animal elements. But of course, we eat the food that we are growing on cropland. And so often, as here in New Zealand, that food is deficient in these essential elements like selenium. So both cropping and pasture land should be tested for all the animal essential elements as well as the plant essential and beneficial elements to make sure food has a full complement of all the nutrients that we humans need. There's some other special examples here. The bacteria that fix the nitrogen that live in symbiosis with legumes need cobalt or vanadium. This is the key element that's at the heart of the nitrogenase enzyme that is responsible for fixing nitrogen. So if a soil is short of cobalt and vanadium, you can have legumes growing there, but they won't be fixing any nitrogen. They'll just be using soil nitrogen. There's also iron, sulfur, and nitrogen in the nitrogenase enzyme. So if any of those are deficient, nitrogen fixation may not be working either. Nitrogen fixation also uses a lot of energy. Nitrogen fixation is the process of ripping dinitrogen apart and ripping those three chemical bonds nitrogen has apart, and that takes a lot of energy. So legumes need more phosphorus, which is the key ingredient in ATP, adenine triphosphate, than other plants. And if you want to check if legumes are fixing nitrogen or not, dig up some roots, find some nodules, cut them open and see how red or pink they are. If there's white, there's no fixation, and the redder, the more fixation going on. And this red color comes from hemoglobin, exactly the same as the hemoglobin in our blood, whose job is to move oxygen around our bodies. But the nitrogenase enzyme is poisoned by oxygen. Nitrogenase evolved before there was oxygen in the atmosphere. So it evolved in times of anaerobic conditions across the entire planet. And so the job of the hemoglobin is to mop up the oxygen and keep it away from the nitrogenase enzyme. And the bacteria increase the amount of hemoglobin, the more nitrogen fixing they want to do. So it's a very simple way to measure the amount of nitrogen fixation going on. And coming back to these proportions of nutrients in plants and the carbon, oxygen, and hydrogen coming out of the atmosphere, I class these as atmospheric nutrients because they form gases and a major part of their planetary cycles are via the atmosphere. Whereas phosphorus, calcium, etc., representing about 3% of plant matter, I call the lithospheric nutrients because they don't form gases. So they can't cycle via the atmosphere. They can only cycle via the rocks and the soil of the lithosphere, hence lithospheric nutrients. There is a small exception here for sulfur that forms a gas, sulfur dioxide, but the amount of sulfur dioxide produced and cycled by the atmosphere is very small compared to the actual amount of sulfur being removed from soil in harvest, and therefore it's completely incapable of replacing the sulfur being removed. So it has to go on. So sulfur is just treated as a lithospheric nutrient. And then we have nitrogen. I call it the oddball, the weirdo nutrient, because it lives in both worlds, and we'll come on to that in a second. And thinking about nutrients and nutrient cycles as atmospheric and lithospheric nutrients starts explaining a whole load of stuff. Gives you a new perspective. First up, if there were no atmospheric nutrients, if life was made entirely of lithospheric nutrients, we would have to apply the same amount of weight of fertilizer as the dry weight of harvest, whereas 96% of the weight of plants and animals, 90% for animals, cycles freely via the atmosphere. These fertilizers are supplied to us at no cost by atmospheric processes. So weirdo nitrogen, the oddball. Nitrogen sits in both the atmospheric and the lithospheric worlds. Virtually all the nitrogen on planet Earth is in the atmosphere. Indeed, the atmosphere is basically nitrogen. It's just shy of 80% nitrogen. But it's in the form of dinitrogen gas, N2, two nitrogen atoms locked to each other. And this is almost completely chemically unreactive. It's almost as unreactive as a noble gas. So this dinitrogen has to be converted into reactive nitrogen, N(r), to be able to be used by life. So the process of transforming dinitrogen to reactive nitrogen is nitrogen fixation or nitrification. Generally in farming, we talk about nitrogen fixation. And the reverse process transforming reactive nitrogen to dinitrogen is denitrification. So all the nitrogen in living things and dead things originates from the atmosphere and eventually returns to the atmosphere. Because dinitrogen is so chemically unreactive, there is no nitrogen in the rocks of the earth. Literally, all the nitrogen is sitting above our heads. There is no nitrogen worth talking about underneath our feet. So I'm going to step you through a reasonably comprehensive, from a farming growing perspective, nitrogen cycle. It gets way more complicated than this in the details, but those details are not important in terms of farming and growing. And understanding the nitrogen cycle is going to help you better manage nitrogen, save you having to buy so much fertilizer, and also reduce the potential for having nitrogen leaching into the environment, and causing pollution. So we start up at the top in the atmosphere with dinitrogen gas. That is fixed by bacteria and archaea. These are called diazotrophs. Diazotroph is an organism that fixes nitrogen, and they are all bacteria and archaea. And they live both freely in the soil and in symbiosis with plants, particularly the legumes. They are the most important in agriculture, but there's other species. Older trees fix nitrogen. Gunnera, the dinosaur rhubarb, also fixes nitrogen. And we have nitrogen fixation as part of nitrogen fertilizer production. It is exactly the same chemical transformation, the same chemistry in both the biological and the synthetic nitrogen fixation. Indeed, for decades now, half the nitrogen that is fixed on planet Earth is fixed by synthetic factory-based production. So half the nitrogen in your body was made in a factory, not on a farm. Cyanobacteria there, commonly called blue-green algae, they are not algae, they are actually bacteria that photosynthesize. The first reactive nitrogen chemical made is ammonia, NH3. So you take your dinitrogen with its three chemical bonds, you rip those apart and stick a hydrogen atom, our simplest chemical element, onto each of those chemical bonds. Ammonia is a gas, so it's volatile. It is used as a fertilizer injected into the soil in North America. Few other places actually do that. So bacteria and archaea transform it into ammonium, NH4 plus. Now, a key thing here is virtually all these transformations of different forms of nitrogen from one to the other in soil are being done by bacteria and archaea. They are doing the heavy lifting here. Ammonia NH4 has a positive charge on it, a plus. So that means it binds to soil particles. It is a cation. And ammonia is one of the two forms of nitrogen that can be assimilated, taken up by plants. It's also assimilated by microbes, bacteria, fungi, etc. Once it's been assimilated by plants, taken up by plants, it is by de facto no longer in a mineral form. Ammonia and ammonium are mineral forms of nitrogen, inorganic forms. There's no carbon in there. As soon as it enters biology, ipso facto, it is in an organic form, as in organic chemistry, the chemistry based around carbon compounds. And then plants shed leaves, they die, they get eaten. The animals that eat them urinate, defecate, they die as well. And that reactive organic carbon ends up in the soil as soil organic matter. And more than 90% of the total nitrogen in soil is in the form of soil organic matter. And most of the mineral forms of nitrogen, the ammonium, the ammonia, are actually attached, sorbed onto that organic matter as well. So the vast majority of soil nitrogen is in the form or on organic matter. The thing is, plants can't take up nitrogen or any other nutrients in the organic form as organic matter because the molecules are simply too big to go through the root barrier, the surface of the root, and there are not the biochemical uptake pathways that can latch on to organic molecules and pull them into the plant. For each and every nutrient, there is only a small one or a small range of mineral inorganic forms that plants can actually take up. So that organic matter has to be decomposed, mineralized back to inorganic mineral forms before it can be taken up by plants again. And this is the process called ammonification. Organic matter is converted back into ammonium. And this is where the fungi appear, and you can see the little fly agaric there. Fungi are actually really complex multicellular organisms compared to bacteria and archaea. One of their key roles is decomposition. One of the reasons we have so much coal from the Carboniferous period is not only was it nice and warm and wet and full of marshes and stuff, plants had evolved lignin, which is what makes wood wood. Lignin is simply a carbon polymer, just like plastic is a carbon polymer. Plants evolved lignin, but there was nothing in existence that could decompose lignin, just like biology cannot decompose the plastics we've made. After a while, so the trees died and simply just did not decompose. They just lay there and built up and built up to form the massive coal seams we have today. But then the white rot fungi evolved a way of decomposing lignin and basically stopped the build-up of those dead trees by decomposing them back to carbon, oxygen, hydrogen, returning to the atmosphere, and stopped the coal measures being built. So fungi speciality is being able to decompose, to rip apart complex chemistry that nothing else can. And indeed, now fungi are starting to evolve and are being bred to be able to decompose plastic. It's just a carbon polymer, nothing particularly fancy about it. Now, if the nitrogen cycle ended here, everything would be sweetness and light. We would have no problems associated with nitrogen, but unfortunately, it carries on. So the ammonium is transformed by bacteria and archaea into nitrites, which is unstable, and then into nitrates, which are stable. NO3 minus. Nitrates are the other form of mineral nitrogen that can be taken up by plants, and it requires less energy to do so. So plants often take up nitrates in preference to ammonium. But that little minus sign on nitrates is where the trouble starts. Nitrates means the minus sign means it has a negative charge. So unlike ammonium, which binds to soil, nitrates don't. So if there is drainage, nitrates will leach out of soil. They are washed out of the soil. Nitrates are also the main form of soil nitrogen that's formed by abiotic, non-biological processes. Depending where you live and the amount of lightning that occurs, a few kilos to a few tens of kilos a year of nitrogen is deposited from nitrogen, reactive nitrogen generated by the very high pressures and temperatures in lightning. Most of that nitrogen is in the form of nitrates, a little bit is in the form of ammonium as well. But generally, compared to the rest of the flows in the nitrogen cycle, it is a relatively small amount. Natural ecosystems, it's plenty to replenish nitrogen that's lost, but not in farming. Nitrate is also the main form of mineral nitrogen that's denitrified by bacteria and archaea and returned to the atmosphere as dinitrogen gas. But worst of all, nitrates, when the soil is anaerobic, the bacteria and archaea, and let me just move the picture out of the way here for a second, create a whole load of hideous nitrogen gases that are among them greenhouse gases, ozone depleters, toxic, and causes of acid rains. And those brown hazes you see over large cities, particularly in warmer climates, they are these nitrogen gases. So that is the totality of the nitrogen cycle that you need to know for farming. And what we also have here is essentially two linked nitrogen cycles. Around the outside, we have the inorganic, the mineral nitrogen cycle, nitrogen gas going through various mineral forms in the soil and returning to the atmosphere. And then in the middle, we have the organic nitrogen cycle. Organic matter decomposed, taken up by plants, returned to organic matter, and they're linked through nitrates and ammonification and ammonium. So the fact that soil organic matter has to be mineralized for the reactive nitrogen to become plant available has some very significant impacts in the amount of nitrogen being released from soil. To mineralize soil, microbes need the right temperatures and moistures. And we have this concept of biologic zero, which is between 5 to 10 centigrade, 40 to 50 Fahrenheit, and an average about 7 centigrade and 45 Fahrenheit. And this is related kind of a takeoff on the physical absolute zero of temperature. This is the temperature at which the biochemistry in the microbes is slowed to such a point that it basically stops functioning. So particularly in temperate regions, where we get clear swings between summer and winter, between soil temperatures and soil moistures, this has massive implications for mineralization. Starting in early spring, soil is still getting cold. It's the same as the coldest hours after dawn. The coldest soil temperatures are in spring. The soil is wet, but cold means the microbes cannot function and therefore cannot mineralize soil organic matter. At the same time, coming out of winter, any plants that have been growing will be taking up ammonium and nitrate from the soil and depleting them because it's not being replenished. And if there's been drainage, there would have been nitrate leaching, such that under the right conditions or the wrong conditions, depending on your perspective, there could be virtually no plant available mineral nitrogen, nitrates, and ammonium left in the soil. But the atmosphere is warming up, plants are wanting to grow, but there's no mineral nitrogen in them for the soil. So they typically end up with nitrogen starvation. And this is the point where adding some mineral fertilizer that's immediately available or some organic forms that with plant available nitrogen in it, like slurry or biodigestate, or applying foliar nitrogen fertilizers can fix this problem. As we move into later spring and early summer, the soils are warming up, which is helping with mineralization in there, but they're also tending to dry out. So you can get an increasing amount and then it starts waning again, particularly as you go into summer, autumn. The soils are warm to hot, but typically dry. Typically, soil microbes, indeed a whole load of organisms, including plants, once it starts getting above about 25 centigrade, they start actually performing worse. Some things clearly like it hot, but most things 25 centigrade is kind of starting to get at the top of their preference. Then as we go into autumn, soils are cooling down but getting wetter again. So potentially you're going to see an uptick in mineralization as you go into autumn and the soil starts filling up with ammonium and nitrate again. And then we go into winter, mineralization basically grinds to a halt. The plants start taking up the remaining nitrate and ammonium, and we start getting drainage again. So you start losing nitrate out the bottom. So understanding this is really important to understanding when is best to apply fertilizers and when it's best to rely on soil nitrogen. And then we come to phosphorus and the lithospheric nutrients. So phosphorus here is a proxy for all the lithospheric nutrients, as the cycles at the global scale are fundamentally the same. And so we've got two main phosphorus cycles and lithospheric nutrient cycles. The soil cycle over here on the left. Phosphate in soil as plant available forms is taken up by plants, and then just like nitrogen, they die, they get eaten. The animals then also die, get eaten, and eventually that organic matter and the phosphorus in it, the nutrients in it, returns to the soil organic matter pool, where it's decomposed, turned back into plant available forms and taken up. This can cycle in a matter of days, potentially even hours, but years, decades, centuries. Some phosphorus may get stuck in particular parts, end up as undecomposable material like animal shells, bones, etc. Even under natural ecosystems, some of the phosphorus is lost to rivers and streams, ending up in seas and oceans. There it settles out at the bottom as a sediment that builds up in thickness and eventually gets compressed into new rocks. That in turn, eventually through plate tectonics, gets uplifted into forming new land. That rock is weathered, the phosphates released from the rocks and returns to soil. This is clearly a lot slower than the soil cycle, typically talking tens of millions of years to hundreds to even billions of years for phosphorus that's lost from one piece of soil is returned in the distant future to another piece of soil. For humans, phosphate and the lithospheric nutrients that are lost from the soil cycle into the geological cycle, this is effectively a one-way trip. Indeed, our depletion of phosphorus reserves by putting phosphorus fertilizer on land and then essentially taking the human manure that's produced from that food and tipping it into the oceans is one of the biggest challenges facing agriculture over the longer term. So phosphorus specifically has quite a lot of complexity about it. So, like nitrogen, we're going to take a bit of a deeper dive here. And there's a whole range of things that affect how soil, the availability of soil phosphorus, both the inorganic and the organic solid phases of phosphorus, how much biological activity, the chemistry of the soil, and again, like nitrogen, soil moisture and temperature is also important. So another little walk through the soil phosphorus. This is not so much a cycle as a series of pools. So we have our plant in the center, roots in the soil, leaves in the sky, but phosphorus doesn't form a gas, so there's no atmospheric component here. The phosphorus originates from the rocks that form from which the soil is formed. The principal mineral is apatites. Those rock particles in the soil, the grains of soil, are continuously weathered, attacked by soil chemistry, organic acids, and that releases from the apatites soil solution P. This is orthophosphate, so typically we talk about phosphate in soil, and this is the form that plants can take up. Then there's a communication, an exchange between the soil solution and the secondary phosphorus minerals. These are calcium, iron, and aluminium phosphates. So these get created and destroyed as soil solution. Phosphate in the soil solution binds with calcium, iron, and aluminium in the soil. That's a relatively slow process. There's also sorbed P that's stuck, absorbed, or stuck on the surface of clays and aluminium and iron oxides, and also carbonates. This is a more quick exchange. So as soil solution is depleted, the P that's sorbed onto these soil particles is released. And considering, depending on the parent material, there could be several tons, even tens of tons of phosphorus in the plough layer, the A horizon of the soil, and particularly if you then include the B horizon below it, there may only be a few hundred grams to a kilogram per hectare of soluble P that the plants can take up. And plants are often taking this up at considerable rate, so there is often quite a rapid release of sorbed P and slower release of secondary P minerals into that soil solution. So they're going backwards and forwards depending on the amount in the soil solution. When we add mineral fertilizers like rock phosphate or superphosphate, they go into the soil solution. And what happens is that creates a large amount of soil solution because we're typically putting a significant amount of phosphate fertilizer on. So quite a lot of that applied fertilizer then goes into the sorbed P and the secondary P minerals. So often when you apply phosphate fertilizer, a large amount of it is quite rapidly made plant unavailable. So plants take up um, orthophosphate from the soil solution. We then get losses from the soil phosphorus system through crop offtake and livestock removal, particularly livestock because our bones are made of principally calcium and phosphate. Some of the plants growing, particularly in pasture situations, will leave residues and organic inputs such as manure and compost, etc., return that phosphorus to the organic phosphorus pool, i.e., soil organic matter. Now, we've just talked about the nitrogen soil organic matter pool. There are not different organic matter pools. There is one soil organic matter pool, and because plant material, the material and dead animals, etc., dung, urine that goes into that organic matter pool has a complete range of nutrients in it. The organic matter pool has nitrogen, phosphorus, potassium, calcium, etc. in it. So it's just one organic matter pool producing a whole range of different mineral nutrients as it is decomposed, as it is mineralized. And that's what happens here for organic phosphorus as well. The organic matter is mineralized, the nitrogen is released, the phosphorus is also released, and it goes back into the soil solution pool. The microbes in soil, they're also dipping their hands, so to speak, into the soil solution pool and pulling phosphorus out of that to build themselves up. And then the other main loss routes for phosphorus from the soil system. First, leaching. Phosphorus does leach. It's not a major source of phosphorus leach loss from soil, but it does occur, particularly when there's drainage and via things such as field drains. But the largest loss of phosphorus is with runoff and erosion. Essentially, soil is being washed off the fields, entering waterways, along with some soluble P, the soil solution P, and that's ending up in waterways and is lost from the soil system. Soil phosphorus, soil pH has a very large impact on its availability, so not how much phosphorus is in the soil, but whether plants can access it. At really acid soils, the phosphorus is fixated, locked up by iron. As the pH increases, it then gets locked up by aluminium. And then we have a bit of a sweet spot between pH of about 6 to 7, where phosphorus is the most available. But then as pH increases further, it gets locked up, fixated by calcium. And it's always iron, aluminium, and calcium that are playing the key roles in phosphorus here, also in those phosphorus pools. They are key elements there. So a number of other elements, including micronutrients, are most available in this 6 to 7 range. But as phosphorus is one of the most important nutrients and it's so impacted by pH, this is why we're typically aiming, at least in temperate regions, for a pH of around 6.5 because that maximizes phosphorus availability. And all this complexity is why we've got all these different soil tests, soil P tests, that give us different results, such as Olsen P, resin P, etc. What they're doing is they're measuring slightly different bits of different soil pools. And also your pH strongly impacts plant availability. So a soil nutrient test is very simply just a probability that if you apply a fertilizer, apply a nutrient, whether you will see a plant growth response or not. That's all soil tests are doing. Are you likely to see a plant growth response if you add nutrients? And because of this complexity and these different tools, you can get really quite contrasting results from different soil phosphorus tests, and even have your soil phosphorus by the same test increase even when you haven't added fertilizer because you perhaps increased your soil pH and that's releasing more phosphorus. Next is micronutrients. There is a wide misconception here that micronutrients are not as important as macronutrients because they're required in smaller amounts. But you are just as dead if you have a micronutrient deficiency as if you have a macronutrient deficiency. You are just as dead if you're missing 0.0001% copper as if you're missing 1.5% nitrogen. Prior to the advent of our modern mineral fertilizers, the refined and synthesized fertilizers, most nutrient sources going onto farm were from organic sources, guano, which is seabird manure or fossilized seabird manure and things like compost and manure. These all come from sources that contain a full suite of the macro and micronutrients. But our refined and synthesized fertilizers, stuff like rock phosphate, superphosphate, ammonium nitrate, urea, etc., they contain little or no micronutrients. And what happened as we went from the 1800s into the 1900s and increasingly using these refined and synthesized fertilizers, the inputs of micronutrients to farms stopped. And at the same time, we're increasingly focusing on the macronutrients pH, the old NPK. What's this resulted in is over the decades, both the global north and global south, because in the global south, typically there's just not enough fertilizer going on full stop, we've ended up with significant micronutrient deficiencies. Because they're only required in small amount, it takes quite a long time, many years, decades even, to draw down the amount of micronutrients into the soil to the point we get into deficiencies, but we are now there. So it's essential to always test for all micronutrients. In more extensive systems, maybe 5 years. For more intensive systems, maybe every couple of years. pH. pH is mercifully simple. You just do soil tests, whether you send it to the lab or use an in-field pH soil testing kit. And if your pH is too low, you add lime. If you've got too high a pH, you probably have some bigger problems and you really need to get expert advice from a soil and fertilizer scientists. The law of the minimum, often called Liebig's law and Liebig's barrel. Liebig, Justus von Liebig, the father of agricultural chemistry, did not actually come up with the concept, but he widely promoted it, and so his name got stuck to it. The metaphor here of the barrel is it's the shortest stave that determines the maximum height of the water in the barrel. So ditto for the nutrients. It is the nutrient that is in the shortest supply, macro or micronutrient, that determines plant growth. It's the limiting nutrient. This is a direct consequence of stoichiometry. So this is another reason for thinking about nutrients from this perspective. So the message here is all the staves need to be all at full height for the nutrients to be at optimum, for the water level to be highest, for plant growth to be the best, and soil biology to be the most healthy as well, and therefore overall soil health to be the most healthy. Now, as this report and presentation originated in an organic agricultural context, I do want to have a look at the organic nutrient sources here. So this is stuff like manure, plant residues, compost, biodigestate, etc. A key challenge is if your soil is like the barrel in that previous picture where it's got very unequal staves, you can't fix the stave that's too short without oversupplying the stave that's too long, the nutrient that's deficient with a nutrient that's current at optimum, using organic nutrient sources because you can't control the proportion of nutrients in these materials. Just like the living things this originated from, these organic nutrient sources are mostly carbon, oxygen, hydrogen, and with a smattering of nitrogen, phosphorus, potassium, etc. So if you have unbalanced nutrients in your soil, like the staves on that barrel, you need to use straight fertilizers, mineral fertilizers, so you can correct the deficient nutrients up to optimum levels without over fertilizing the rest. And indeed, as I've touched on before, excessive levels of nutrients are becoming an increasing problem in terms of pollution, but also not fairly sharing nutrients around. And there's a particular problem here with intensive horticultural operations, particularly the really intensive organic market gardens, massively over fertilizing with compost. Compost should only ever be applied to replace the nutrients that have been lost in harvest. If you want to picture what that looks like, think of taking your harvest and instead of selling it, composting it. And how much compost would be produced. That is replacement. On a slightly more practical level, light feeders, stuff like salads that are literally just leaves full of water, 95% water, a few hundred kilos per hectare or pounds per acre per year would be more than sufficient to replace the nutrients that are being removed. Heavier feeders, stuff like potatoes, might require 30 tons a hectare, 12 tons per acre to replace the potassium and phosphorus that's being removed. To give you a visual idea of what that looks like, if you apply 30 tons of compost to cultivated bare ground, you won't see it if you're standing up. You will have to get down on your hands and knees. 100 tons of compost a hectare will just cover the soil. You'll still see some bits of soil crumbs poking through in places. But these intensive organic horticultural operations, often compost is being used as a mulch. Centimeters, inches are put on the soil, and that is centuries, even millennia worth of nutrient supply, particularly phosphorus and potassium. This is now completely unacceptable. It may be permitted in organic systems, but this has to stop. And compost itself is actually a really poor source of nitrogen because between 25 and 75% and potentially even more of the nitrogen is lost during composting. And this is going back to our stoichiometry and our lithospheric and atmospheric nutrients. Because what's going on in composting is the atmospheric nutrients, the carbon, oxygen, hydrogen, and nitrogen, are being lost, being returned to the atmosphere. Nitrogen is lost as ammonia, And also it's denitrified back to dinitrogen gas. That's where the nitrogen goes. Whereas the lithospheric nutrients don't have those gaseous forms, so they can't escape from the compost heap. And so their total amount doesn't go up. There's the same number of atoms in the starting material as the compost, but their concentration has gone up because the majority of the biomass, the carbon, oxygen, hydrogen, and a whole chunk of nitrogen, have been returned to the atmosphere in the composting process. Whereas anaerobic biodigestion takes place in anaerobic conditions in a sealed container. Indeed, anaerobic biodigestion is directly comparable to what happens in the rumen, the digestive tract of herbivores. They are literally giant steel rumens. Because they are a sealed container, none of the nitrogen can escape. The only thing that comes off a biodigester is methane, which is just carbon and hydrogen, CH4. And what happens is the product of anaerobic biodigestion is basically the same as a ruminant's digestive tract. It's slurry. It's literally like cattle slurry. And it has a considerable amount of the nitrogen, not in the organic forms, but in the mineral forms, as nitrate and ammonia. And therefore, it's immediately plant available. And a really good example of the benefits of anaerobic biodigestion versus composting was an organic stockless system, not directly contrasting composting. But they had an organic stockless system. They had a pasture phase to increase soil nitrogen and soil health. And they either mowed and dropped the pasture in place, or they mowed the pasture, harvested it, took it to the anaerobic biodigester, digested it, and then took the slurry and put that onto the cereal crops. That increased yield by 27 to 47% due to far better nitrogen utilization than a drop in place or a composting system would produce the same results as the mow and drop system. And finally, the third soil health, biological soil health. This is all about the biodiversity of soil organisms and their ecology. Ecology is the science of how organisms interact with each other in their non-living environments. In this case, soil. And as I've highlighted several times, soil is the most biodiverse ecosystem on the earth. And virtually all of that biodiversity, especially the genetic biodiversity, is exclusively bacterial and archaea. And here we have a genetic biodiversity diagram. Up the top here in the dashed circle, we have the bacteria. Down here, we have the archaea. And over here are the eukaryotes, everything that's not a bacteria and archaea. And you can clearly see the bacteria in particular dwarf the genetic diversity of the eukaryotes. We, as animals, Animalia, do not even get a name check on this chart. That's how little genetic diversity there is amongst the eukaryotes. We are virtually all genetically identical, even though we look dramatically different. And protista, things like amoebae, all the multicellular organisms are in the eukaryotes, the fungi. We are far more closely related to fungi than either fungi or us is related to bacteria. And as this tree implies, it shows that the eukaryotes have evolved from archaea, not from bacteria. Indeed, it is when an archaea swallowed a bacteria that became our mitochondria that the eukaryotes evolved. So the people the organisms in terms of soil biology that are important are plants, bacteria, and archaea with a bit of support from the fungi. These are the important organisms in soil. Everybody else is to mostly a bit walk on part. And soil biology and ecology reflecting this are now principally using genetic techniques and processes measuring enzyme activity. So they just go in and measure the genetics in soil to work out who's here. Because most of these bacteria and archaea and a whole load of the fungi cannot be cultured in labs. The only way to measure these things is through genetic processes. So I'm going to come back to this, but a lot of the commercial soil biology analysis services are typically still using optical microscopes for measuring things such as nematodes and amoeba, protista. It's completely irrelevant. So, what does this new soil science revolution, POM and MAOM, tell us about how we should be managing soil biology? It's maximizing the diversity of living roots year round. It's on the basis of good physical and chemical health. I have to say, this is something of an anticlimax. That's it. That's all I've got to tell you. There's no complexity. You don't need to do complicated things with brewing stuff up. Maximizing the diversity of living roots year round. That's going to maximize your soil biology. It's a bit dull and boring, I have to say. And so we return back to this measuring soil biology. So there's a whole range and growing number of commercial soil tests that are being offered. Um, but the key organisms that are driving soil biology and soil health are plants, bacteria, and archaea, and some fungi. And they are mostly not measuring your plants. You know what plants you've already got in your on your fields. They may be estimating how many bacteria and archaea, but these are really small and you've got no way of differentiating these things, most of them under a microscope. You can only do it through genetic testing. So most commercial providers do not use these tests. Some of these tests used by soil geneticists cost thousands, if not tens of thousands of dollars per sample. These are very expensive activities being undertaken here. So, as these, I'm assuming farmers and growers are not paying that kind of money for an individual soil test, and most of these commercial providers are not offering them. So, these commercial providers are simply not measuring the right things in soil. But even if you did pay thousands of dollars to get an estimate of all the different organisms, principally bacteria, archaea, and fungi in your soil, and you have that information, can you do anything with it? I think that for information to be valuable to a farmer or grower, you have to be able to take a management decision on it. And you simply can't take any management decisions on the result of a list of species in your soil. There's lots of value to scientists as they try and pick this stuff apart, but not farmers and growers. And I think a lot of confusion has arisen here because of the contrast with soil nutrient tests. We've been picking apart soil nutrients since the end of the 1800s, if not before. And we have a vast amount of science now, and it shows there's a simple linear direct causal relation between nutrient levels and plant growth. Plants are made of nutrients. If there's deficiency in a nutrient, Liebig's barrel, if you add that nutrient, the plant will grow more. It's a simple linear causal relationship. We now have more than sufficient science understanding the complexities of the soil ecosystem and soil biology to know that it's insanely complicated. Virtually all of it is non-linear. Much of it is feedback loops with chaotic outcomes, such as there is no simple cause and link between even kingdoms of organisms. This fungal bacterial ratio that some people are interested in. Now, fungi are a kingdom, bacteria are a kingdom of organisms. You simply cannot look at that and say, okay, what management decision should I do here to improve plant growth beyond a diversity of living roots year round? What we do know is up to a point, having more biology and having more active biology is better than less. What's one way of measuring overall biology? Well, it's the DIY soil test that I'm going to be telling you about shortly. And the advantage of these things is as you are doing them, you are learning about soil by doing them. If you do want to get a lab test done, the best option is what's called the hot water extractable carbon test, or similar kind of terminology. Unfortunately, it's a really terrible name because it describes the method used in the test, not its purpose. A soil phosphorus test tests for soil phosphorus. It's self-explanatory. It does what it says on the tin. What a hot water extractable carbon test is a proxy measure for total microbial biomass, i.e. soil biology. As the name explains, you get a small sample of soil, soak it in hot water for about 24 hours and measure the soluble carbon. So soluble carbon is things like sugar, what you put in your tea and coffee. it's a soluble carbon compound and proteins and oils and stuff. Microbial biomass is mostly made of these soluble types of compounds. So that is why it's this very good proxy measure of total microbial biomass, which is very expensive to measure. We are talking thousands of dollars. And as it's a very simple, inexpensive test to do, it doesn't cost very much, a few tens of dollars. Get it done with along with your soil nutrient test to give you a numerical value for your soil microbial biomass so you can track it over time. Microbial biomass also very rapidly responds to management changes compared to total soil organic matter. So, as I've touched on before, the cutting edge of science is moving on from this static snapshot of species to studying the dynamic processes. As I've said before, a dead body, a newly dead body, and a live body have the same nutrients inside them. It's the dynamic processes within inside living things that make things alive. And as soil is now considered to be a living thing in and of itself, it is those dynamic processes that are the most important. So again, going back to those commercial soil biology tests, they're just taking snapshots. They're not telling you anything about the processes going on in soil, such as the oxidation of ammonium to nitrate. So anybody that's talking about bacterial to fungal ratios or nematodes and protists in the soil is living in the last millennium. Moving on. So what do you need to do in terms of farm management to improve your soil biology? Well, for pastures, the first thing is we need to increase the biodiversity in the pasture. We need several species of each of the grasses, the legumes, and the forbs, which is basically everything's not a grass and a legume, to increase that plant diversity, to increase the diversity of exudates. And then it needs to be managed through rotational grazing, not set stocking. Set stocking is a colossal disaster for livestock management. This is where stock are basically just spread across the whole farm and allowed to eat whatever they want. What it does is it results in the stock moving around a lot, looking for stuff they want to eat because they've eaten out all the palatable species, the tasty stuff. The pasture is invariably short, so there's low photosynthesis going on, which means low root exudates, there's low feed production and low profits. Indeed, set stocking is just so bad. Don't do it. Rotational pasture management is where pasture is allowed to grow high, typically around 30 centimeters a foot. Stock are put on just long enough for them to eat it down to the required residuals, the required height, and then moved on somewhere else, either to fresh pasture or to standing off areas. And then the pasture is allowed to regrow, typically 2, 4, 6 weeks, depending on the time of year and your climate and everything else. What this does is by maximizing the biomass, you're also maximizing the leaf area. So you're going to maximize photosynthesis, exudates, the amount of feed you produce, the livestock performance and profit. So basically, there's everything to like about rotational pasture management, apart from the extra work involved. As the stock really just offered a huge buffet, they tend to eat everything, the palatable species and the less palatable species, which is principally grasses, along with unpalatable species like docks that are actually edible but just don't taste very nice. So you get much better weed control in a diverse pasture and a rotational pasture management because the extra species fill in the ecological niches the weeds would otherwise occupy, and the weeds that do get in there, unless they're completely inedible, will get eaten along with everything else. Rotational pasture management or rotational grazing is also key to managing stock in wet weather to avoid that compaction, that squishing of the macropores. And this includes fodder crop, particularly if the fodder crop is a multi-species fodder crop. So the stock go onto the pasture. It's a diverse range of species. It's more palatable them, so that they wander around looking for stuff to eat. They will just go in and start eating immediately. They will eat rapidly because they're hungry. You put them on hungry, and as soon as they're full and they want to start ruminating, you pull them off again and put them somewhere where they're not going to cause soil compaction, like a standoff pad or a farm lane or a barn or whatever. What happens with this approach is you get the minimal amount of stock walking around, moving, and then you minimize the compaction. They go on, they eat, they come off. the least amount of walking around. Increasing plant diversity in cropping systems is somewhat more tricky than in a pasture system. For me, cover crops here are the easy win. Start filling in times when soil would other be bare with diverse multi-species cover crops. Even if it's going to be bare for just a few weeks, put a multi-species cover crop in. And a lot of people have looked at this and kind of gone, wow, that's a colossal waste of seed, isn't it? Well, research in New Zealand has found statistically significant increases in soil aggregation, which again is that really good proxy measure for overall soil health over these really short periods when using these diverse cover crops. Then to diversify the cropping part of the system, we need to get into intercropping, which is simply growing two or more plant species together, i.e. not monoculture. So this gives us diversity in space. And this is where things get complicated really quite quickly, and the potential for things to go badly wrong is quite considerable. So do not bet the farm on this stuff. Start small. So we got mixtures of cash crops such as a cereal like wheat and a legume like a pea or a bean. These are a really great, a really good intercrops is mixing a grass and a legume together. That's the basis of pasture in New Zealand. We grow ryegrass and white clover as an intercrop here, even though it's not widely recognized as an intercrop. You can mix cash and what I call service crops, cover crops. Service here is as in ecosystem services. These things are providing a service to the farm system. For example, clover as a living mulch under maize/corn. There's a whole FFC report done a couple of years ago on service crops and intercropping, and there's a link in the main report and the description below. And then you have rotations, which are plant diversity over time. Lots of research that shows that as you increase plant diversity over time, you improve soil health. And there's a common misunderstanding about the value of annual cover crops versus perennial vegetation, i.e. pasture herbaceous species. A lot of people are in the situation of ripping up pasture, for example, between vineyard rows, or even worse, whole paddocks of pasture to plant annual cover crops. It is completely the wrong thing to do, and it is far worse for soil health. The thing with annuals is their life strategy is to be a seed. They are typically a seed for multiple years. They are a plant typically only for a few months. So they need to produce lots of seeds. So most of their biomass, around two-thirds of their biomass, goes into their above ground foliage, which then is funneled into the seeds. So only one-third of the biomass is in the form of roots. And because they only live for a few months, they're not really needing to get so much support from the microbes in their rhizosphere. They're kind of like the fly-by-nighters of the plant world in terms of microbes. So they don't produce as much root exudates. So they've got small root systems producing small amounts of root exudates. And it's root exudates that drive the whole soil ecology and soil health. And indeed, this is why annual cropping systems, arable and vegetables, have much lower soil organic matter and soil health than pasture-based systems, not because of the tillage, the cultivation, but because they are growing annual crops which don't produce very many root exudates. In comparison, perennials, particularly a pasture species, their life strategy is to be a plant, and they may live in the same place for years, decades, centuries even. And particularly for the pasture species that have evolved to survive grazing, defoliation, they put two-thirds of their biomass into their roots so they can recover from being grazed, having their heads cut off. And because they're in that same place continuously for many years, they really want to look after their microbial mates, so their microbes look after them. So not only have they got two-thirds of their roots, their biomass as roots, they're producing lots of root exudates to look after. So they are far more effective at building soil organic matter, principally or mineral associated organic matter, than annual cover crops ever will do. They're also in the ground for far longer. This is why pasture is so effective at increasing soil organic matter, not because there's a lack of cultivation, but because it's perennial plants growing out there. As part of all this new understanding of soils and soil organic matter formation and the importance of soil health, there's increasing interest in the use of biofertilizers and biostimulants. Now, biofertilizer is very simply any biological, i.e., organic chemistry material that has sufficient plant nutrients in it to achieve a plant growth response if those nutrients are deficient. Typically, biofertilizers are thought of as stuff in bottles that you buy from the farm supply store, but manure and compost are biological materials that supply plant nutrients. So they are also technically a biofertilizer. But I'm principally talking here about these things in bottles you buy with money. Biostimulants also come in bottles and they are also biological materials, but typically they don't contain very many plant nutrients. Clearly, they have to contain something because there has to be a biological compound in there, which means there has to be carbon, oxygen, and hydrogen in there at the very least, if not some NPK as well. What they do is they produce a plant growth response because they interact with the plant's biochemistry and stimulate it to grow more. And probably the classic example of this is gibberellic acid. It's a plant auxin, a plant enzyme that promotes vegetative growth. Some seaweeds contain quite good, significant quantities of gibberellic acid, which through processing can be concentrated. There's also synthetic forms of gibberellic acid, a product in New Zealand called ProGib. If you apply these to example pasture, you will get a plant growth response, not because you applied any nutrients worth talking about, but because you stimulated plant biochemistry. And then we run into the problems. Some of these biostimulants and biofertilizers are backed by solid science. They were produced through extensive research programs. Others have no science supporting them at all. Some of them, science says they do not work. And particularly around soil health, many of these products are sold that they will enhance soil health. But if you extracted the biology in reasonably healthy soil from 1 hectare of farmland, you would end up with a cube of biology when you've essentially wrung the water out of it, a cube of 30 cubic meters, which is a bit bigger than 3 by 3 by 3 meters, so kind of nearly 4 by 4 by 4 yards. Most of these products, again, if you wrung all the water out of them, would add a few hundred milliliters or a few liters, a few pints of microbes to that hectare of farmland soil. Clearly, if you had a 3 by 3 meter cube of microbes and you tip 100 mils or a few pints of microbes into that 30 cubic meters, they are kind of up against it. In addition, when the microbes are in the soil instead of a big old funny cube, they're in a complex interacting ecosystems, and ecosystems are very good at resisting invasion by external entities, organisms coming into them. So very simply looking at this example here, what is the chance of these added microbes or biostimulants and biofertilizers of having any effect on soil biology and the soil ecosystem? The answer is, of course, very vanishingly small. In comparison, where there's a healthy bunch of diverse plants growing in that same field, they are potentially producing hundreds of kilograms to tons of root exudates per hectare per year. And the microbes and the plants have co-evolved together. So exudates are the perfect microbe food. There is simply nothing you can apply to soil in the kind of quantities that are going to come anywhere close to boosting health when it comes to root exudates. Once again, it is a diversity of living plant roots year round that drives soil health and soil biology. If you want to dive into biostimulants and biofertilizers, there's a slightly older report here available from the Future Farming Centre and my website as well. And again, QR code will take you directly there. And I will link to that in the description, and it's in the main report as well. So you can think of plants as kind of like a zipper that joins the sky and the soil into a single continuum. They're pulling the carbon dioxide and oxygen and taking the water out of the atmosphere and pumping it into the soil to drive soil biology and pumping that energy in there as well. So it's literally like this picture of a tree here. It's like the links in a zipper holding the sky and the soil together. And finally, DIY soil health tests. So these are tests you can do it yourself in the field. Indeed, you can only do most of them in the field. So we're going to have a look at all of them. And again, QR code for the main report focused just on DIY soil tests from the QR code in the report and the description as well. This is just going to look at these ones listed here so you know about them. I'm not going to give you a detailed explanation of how to do them. That's in the main report. A key thing with these DIY soil tests is to compare what I call the middle of the field with the fence line. So the middle of the field doesn't have to be the dead center. It's just the main area of the field where you're cropping, generally avoiding the headlands and gateways where there's extra compaction, with the fence line, which is in some cases, particularly here in New Zealand, we have wire fences. So it's right under the wires, or it's just on the other side of the wires if that's uncropped ground. But it's some soil close to the main field that isn't being farmed, but otherwise has nutrients, fertilizers, irrigation, similar to what's going on in the middle of the field. The differences between the two shows you the impact of the farming system on soil health for that specific soil, taking away the variability of soil textures. So first up is the spade test. It's literally just dig a spade-sized hole and have a look. And the first bit's clearly easy. The second bit is interpreting what you're seeing, and that's going to take a bit more experience. But you're looking at things such as the structure, aggregations, whether there's mottling, what it smells like with worms. And once you understand these things very quickly, you will be able to take a spade test and go, this is looking good and this is looking horrible, which is what I showed you before. This nice healthy soil on the left, I'll just look at that and go, that's looking really good. And I'd look at the one on the right. Visual soil assessment, and these have been developed in multiple countries across the globe, and we have one that was developed here in New Zealand, is essentially the spade test taken to its logical conclusion. It undertakes a wide range of measurements by digging up soil and looking at it. There are pictures and scoring systems that are combined with weights to give you an overall measure of your health, your soil's health, and that is comparable amongst different soils and different textures and all the rest of it. This is quite a lot more involved. It can often take 10, 20 minutes to undertake a complete assessment for an individual site within a field. But this is valuable to get into because this will also give you the information to understand what you need to look for when you are just doing a bog standard soil test and digging up a spit of soil. Next to soil probes and penetrometers, a probe same as a tile drain probe. It's just a metal rod about a meter yard long with a handle that you push into the soil. And a penetrometer is basically the same thing with a dial on it, so you can get a measurement of how hard you're pushing. I personally think the probe is the more useful tool. First, it's cheaper. Two, it's almost indestructible unless you drive over it, where plenty of people have broken their dials by dropping them over the years. And actually using your tactile listening, feeling what's going on, I think get much better feedback when you're using a probe than a penetrometer when you're just looking at the dial. The other thing with probes and penetrometers is it only takes a few seconds to push them into the ground, whereas it takes a minute or two to do a spade test and dig a hole up. It's a bit more of a physical exercise. It's also much easier to get deeper into the soil profile than a spade test. These things will go a meter deep. A meter deep hole is a very big hole. I've certainly had these on good soils. I've been able to push these into the ground till the handle hits the soil surface, or my knuckles hit the soil surface. Ring infiltration test measures how fast water infiltrates into the soil, as the name says. Fundamentally, it's a physical health measure. You're measuring that porosity or if there's compaction. But that's all strongly influenced biology, the three-legged stool. So again, it's a really useful overall measure of soil health. And it's pretty straightforward. You get a plastic or a metal ring between about 10 centimeters, 4 inches wide to 30 centimeters 1 foot wide. The soil scientists use bigger stuff, but that's impractical for a farm level. It's about the same height. You drive it into the soil a few centimeters, an inch deep, you fill it full of water, and then measure how long the water takes to go into the ground. That directly relates, therefore, to the rate of rain or irrigation the soil can absorb. E.g., if you've got 100 millimeters or 4 inches an hour in your infiltration ring, that's the rate of rainfall, which is a lot of rainfall, that your soil can absorb. Compare that with 10 millimeters or half an inch an hour. Clearly, you can have 10 millimeters, half an inch of rain in an hour in a heavy thunderstorm, at which point that soil will not be able to absorb that water. It won't be able to infiltrate in, and it will start running off, causing erosion and loss of your soil, your primary capital asset. Finally, slate test for aggregate stability. I've mentioned the importance of aggregate stability multiple times through this presentation. You get small pieces of soil. They have to be dried. If they're not dried, they need to be allowed to dry slowly. So put on a shelf somewhere out of direct sunlight and not force heated. The classic approach to this is get a water-filled jar so you can see through it. You put a wire basket in the top, a mesh of some description. You put the soil in, and it either holds together, so you've got good aggregate stability, or it falls to bits. And in some examples on YouTube videos, again linked to in that main DIY soil health test report, literally it just falls through the basket instantaneously. The aggregates just collapse there and then. And there's an app for this, thanks to the Soil Health Institute in North America. They've paid the University of Sydney to update the Slates app, so you can use this to get a precise numerical measurement of your aggregate stability. And don't just use one test. These tests are like different windows into soil health. Sometimes they can be contradictory. I've often done soil probe tests where it goes in really easily, and then done an ring infiltrometer test, and there's virtually no infiltration whatsoever. So using multiple tests give you all these different views onto your soil health and allow you to build a bigger composite picture. And they're also using different parts of your sensory system, sight, smell, etc. For me, getting tactile, using your senses to interrogate your soil health will help you learn so much about soil and how it functions. So, the importance of soil cannot be overstated for farming, civilization, and our planetary life support systems. But despite being the most complicated, biodiverse ecosystem on the planet and the most frustrating to actually research, because despite the fact it's under our feet, it's completely opaque to normal light. And as soon as we try and measure stuff, we disturb it and disturb the thing we're trying to measure. Soil science is a really challenging thing to undertake. But despite all that, fundamentals of good soil management are really straightforward. Minimize compaction and soil structural damage from livestock machinery and tillage. Make sure all your nutrients, both beneficial and essential, and pH are at optimum, and maximize the diversity of plant species through space and time, along with maximizing photosynthesis to maximize root exudates. That's it. It's that simple. So thank you very much. The QR code to download the full report again, also in the description below, and look forward to seeing you next time.